Just to introduce myself, my name is Vance, and I'm a research scientist and tree breeder with the BC Ministry of Forests, Lands, Natural Resource Operations, and Rural Development. Some people were calling us the Ministry of Everything, but that was already taken by the Ministry of Environment, MOE. So maybe they'll shorten it someday. So I just started here in 2018, and I'm primarily focused on developing 2 new programs for ponderosa pine and interior western red cedar. But I did do my PhD in population genetics, and so it's an honor to be here to moderate such an interesting session with such diverse talks representing all of North America and such different species. So with that, I'm just going to look in the Q&A if you want to submit your questions there. I should also announce that Thomas Parchman is here to replace Lanie Galland. Unfortunately, she wasn't able to make it at the last minute, and so he has graciously agreed to step in and answer any of your burning questions about spatial genetic structure in the California closed pines. So don't hesitate to ask those.
Lanie had she has a couple times a year, she has problems with severe migraines, and so she's essentially unconscious in the dark right now, apparently. So I can try to answer questions on her stuff.
We appreciate that, and hopefully she feels better soon.
Yeah, hopefully.
Yeah. Okay, so I have no questions in the Q&A yet, so I guess I'm going to be starting out with some of my own and just going in order, I guess. I was really interested, Emily, with your hypothesis that there was a glacial refuge right next to the ice sheet in, was it New Brunswick? And I just wanted to ask, this is purely out of my own curiosity, because I've thought about this before for more cold adapted species. I was really curious what cold tolerance is like in butternut.
Yeah, and specifically, I actually just discussed this with someone. It's one of the most cold tolerant species, I think, in its like family or even the walnut group. And so there's actually predictions that because it is currently like its range is much higher in latitude, like goes much farther into Canada than most of its close relatives, that it would be like most likely to be able to persist next to glaciers. And then also then, likely be most influenced by glacial movements in the past. So it is like extremely cold tolerant.
Good to know. Okay, so we have a question here for Kathryn. And maybe this is a little bit more in depth than you were hoping for, Kathryn, but Beth would really like to know more about your path model and if you could explain more about its advantages and why you chose the approach.
No, yeah, this is a great question. I think the major driver for why I used a path model is because initially I used RDAs to kind of parse out these relative influences of, for example, genetic and geographic and environmental on the phytochemical variation. And I was getting a lot of explained variance for both the genetic and the environmental variables on phytochemical variation. And it was almost equal contributions. But I also know that in my system, there's a huge connection between genetic diversity between environmental diversity or variation on genetic variation. So there's a lot of isolation by environment or some local adaptation to environment across my populations. So I wanted to kind of see whether there was actually this indirect relationship of environment through genetics onto phytochemistry. And that path model was allowed me to kind of disentangle that. And so I did find that the majority, at least at the scale I was looking at, the majority of the contribution on phytochemistry was actually genetic. And there was less of an effect of environmental. So yeah, that's a good question.
Okay, thank you, Kathryn. Moving on to Lanie/Thomas. Lionel wrote, I'm excited to hear about the final results. I couldn't help but notice that despite clear clustering on your PCAs, the variance explained by each axis was actually quite low. This would tend to suggest that genome-wide differentiation is relatively low despite clear assignment of some populations into distinct genetic clusters. Do you have any thoughts about that?
Yeah, I think that's a good observation, Lionel. Overall, genome-wide divergence is low. FST between, for example, in Pinus muricata, between the island and mainland.
And this is the
Whoa, I don't know where that came from. Sorry, I had some interfering audio. Differentiation between the island and mainland populations as measured by FST is somewhere around, I think, a 0.1 or 0.12. So yeah, genome-wide differentiation is not pronounced, but this might be, this might be what we expect over relatively recent history, even if these populations are pretty isolated. And I should add that Lanie we just generated this data, where Lanie just started working with this data a couple months ago, so we're still at a really preliminary position in our analyses.
Okay, thanks for that. I'm going to add a question in here for Whitney. You made a very provocative statement in your presentation. You said, bye-bye maple sugar, due of course to the projected contraction of the maple beech forest type in the northeastern United States. However, when I looked at your map, I noticed that it seemed more like a northward range movement into Canada, which wasn't really shown on that map. So I was just wondering if you have any insight into the future of maple syrup production north of the US-Canadian border, for all the Canadians on the call.
Yeah, I was waiting for that question, actually. Good. So thank you for it. So there is an upper threshold, right? We all know that sugar maple, to be for the sap quality and sugar content to be optimal, there needs to be that temperature fluctuation. Well, as it quote unquote migrates and tries to find that suitable habitat, it's going to reach an upper limit as well in Canada. So there is that envelope that would be optimal for it, but it's definitely something that will affect Canada as well. The map that I showed is from the US Forest Service. So I think it was targeting the region where sugar maple is most prevalent. Its range is where it is in the United States. So that's why the Canada was left out. Nothing personal at all. But yeah, it's something that Canada probably should look out for in the future.
Okay, thanks for that, Whitney. It is a really interesting topic. Okay, and then another question for Emily. Zoe would like to know how long genes can be conserved in fossilized pollen. More of a methodological question there.
That's actually something we're kind of discussing now, as if we would actually be able to extract any DNA from fossil pollen. And I don't know if that would be possible. We're also looking into the idea that there's some butternuts that have actually been frozen. And we were wondering if we can maybe get DNA out of them. So yeah, a lot of that stuff I think is still to be tested. It would be really, really fantastic to be able to get some genetic information from there.
Very cool. For Kathryn, a question from Natalie Isabel. Have you observed any tri-hybrids? And to follow up on that, have you observed unidirectional gene flow from one species to the others?
Yeah, so actually most of my hybrids were tri-hybrids, which is, well, yeah, depending on what you consider a species, but I mean, they're hybrids from 3 diverged lineages. So, which was part of the fascinating result of my genetic analysis. Yeah, so hopefully that answers that question. And then as far as unidirectional gene flow, I didn't specifically look at that, though I would be interested in looking in that area in the future. But I know past studies, for example, Terry et al. 2000, looked at that question and found that there was unidirectional gene flow, at least unidirectional integration from the Western species in Washington, Oregon, and California into the Eastern species, Utah juniper. And then that kind of corroborates with what I know about wind patterns also. So yeah.
Thanks, Kathryn. Okay, and then for Lanie and Thomas, this is coming from Tal up in BC. When quantifying diversity, were your estimates standardized in any way? I know Tal's doing similar work, so I have a feeling he has a vested interest in the answer here. It's interesting that your estimates are higher than other conifer species. How do estimates of within population versus between population diversity compare?
That's a good question, and that's maybe something I should have Lanie get in touch with you to talk about more. They're not standardized in any particular way. I know Lanie used methods built into angst to do this, so so that we could incorporate genotype uncertainty, because we have not low sequencing coverage depth, but I would say medium-ish. So we wanted to make sure we accounted for genotype uncertainty when we estimate those diversity metrics. And one of my concerns in making comparisons with other pines and other studies, for example, is that the method that we use to estimate diversity can have a big effect on that. So that's something we clearly need to think about more.
Okay, thanks for that. Susan has sent in a question for Whitney. She's curious to know if you have a good sense of where the zones of hybridization are occurring between Acer species.
I'm so sorry. Could you repeat the last part of the question?
Yeah, do you have any sense of where the zones of hybridization, I think you mentioned it during your talk, where those are occurring between the species?
Well, throughout hard maple range in the US, there's so where there's an area, I don't know if you recall from the range maps that I showed early in the talk, black maple and sugar maple, there's an area where their ranges overlap, their native ranges overlap. And within that, there's specific areas as you go further east. There's you find more sugar maple, but as you go more west, you find black maple. So in between there, maybe around like Southern Ohio, Indiana, maybe Northern Kentucky. Right, there's this pocket where introgression, there's some studies done in like the 1940s and the early 1950s that talk about it. And it's actually, I had to have it translated from French. So hopefully, that's not hard for them to do. But yeah, it's definitely a known area for hybridization where there's some Patrick in that area. Mm-hmm.
I see that Emily is already answering her next question in text. Do you feel comfortable answering it out loud, Emily, or?
Yeah, I
okay.
can go ahead.
Let me ask it first so everyone else knows. So for everyone, Lionel has another question for Emily, and he wants to know about the glaciation versus central marginal hypothesis predictions. Specifically, are butternut refugia what could be considered as the core of the species distribution? He's wondering if it would be possible that glaciation pattern is confounded with the pattern you expect under the central marginal hypothesis.
Yeah, and the question to this is absolutely. The issue with genetic patterns is that, there's not just one evolutionary factor affecting species throughout time, there's multiple. So we could see situations where there was reduced genetic diversity at northern populations and southern populations for completely different reasons, but it would look to us something along the lines of the central marginal hypothesis. The reason why we came up with this idea as being the most likely answer is because we also included that analysis looking at the distance to range edge compared to genetic diversity, and that really showed us a very strong and well-fit linear relationship. So it's likely that, glaciation did have an effect on the genetic diversity of this species with, decreased genetic diversity at northern populations. But we do now see evidence for this sort of spread of genetic diversity really related to the way, to the actual range of the species.
Great, thanks. I'm going to ask one of my own questions to Kathryn really quickly here. I'm just curious what phytochemistry you were analyzing, and I was really interested in knowing, so you said that the Sierra phytochemistry has been preferentially retained in the hybrids. Do you have any idea what advantages that those particular populations would be bringing to the table?
Yeah, that's a great question. So we were primarily looking at terpenoids. So those are can be small and volatile, but we're also looking at larger terpenes too, like sesquiterpenes. Yeah, so I did find that hybrids seem to have a greater overlap in their like constituent compounds with Sierra, and I'm not quite sure why that is or what advantage it could confer. So your guess is as good as mine, but that would be something that I could look into potentially as a really interesting question.
Awesome. Okay. And then for just watching the clock here too, but for Lanie and Thomas, someone here made a comment that interesting study, Pinus radiata is widely planted as an exotic around the world. Did you compare diversity parameters between it in its natural distribution and in areas where it was introduced? And I was actually interested in this because you guys talked about it as a conservation species, and I had to look up where it's planted, and it's New Zealand, Australia, South Africa. So I was really interested in that.
It's actually one of the most widely planted pines in other parts of the world. So it's something that we would like to follow up on at some point in the future, depending on Lanie's future plans. We don't currently have any data for plantation trees in Australia, for example, but it's something we have at least informally discussed. So we don't know anything about it now, but we would like to know more.
Okay. And we're just running out of time here. We might go over, I was given permission to go over a couple of minutes if anyone wants to stay on afterward. But for our official last question, I think we have to go back to the topic of primary interest here. So Whitney, I'm just wondering, you found that Acer nigrum was the most closely related species to Acer saccharum. And I was just wondering if it has potentially any increased stress tolerance and if it could be a viable replacement for a different syrup production industry. I know they make birch syrup up in the Yukon, and even though it's incredibly inefficient, I think it's like 100 to 1. I mean, it's still a luxury good, so there is a market.
Yeah, so I know that there have been selections made based on drought tolerance, and why there is the literature deficit actually exists within this group is because there was an actual common garden study set up in Ohio and in the mid 50s, and they had environmental trials going on with different provenance sources, and then they also had a separate provenance trial looking at sap content, sugar sap content, and quality and yield that they were successful with. So the environmental trials just sort of fell away. And we actually went and were able to like dig through poison ivy and sort of talk to the Seacrest Arboretum in Ohio about the actual, value of this plot that had been left for 60 years. So that was really something fun that I obviously couldn't really talk about today, but so there are definitely environmental advantages for black maple that have been, and they're closely related. So it's been an easy transfer, I guess, or easy to select for.
Okay, great. Thank you. So I think our official session is ending, but there are a couple, if you guys don't mind, if the speakers don't mind, there's a couple more questions that we could potentially answer here. For Thomas, there's one here from Brian Barber. What, if any, phenotypic differences were noted between the coastal pine populations that may explain some of the PCA genetic differences between these populations?
Phenotypic difference?
Yeah, I think just on the islands there, it's like what, 5 kilometers apart. So I think he's wondering if there's anything obvious going on.
So we don't know. I mean, there's been some past work on the radiata down in Mexico. We don't know about the muricata populations. I was pretty surprised to see the genetic signature of differentiation there at those really small scales. When the sampling, Lanie got help. It was during it was the height of the pandemic when we actually had people out collecting some needles for us. So she had some colleagues from the Nature Conservancy that travel out to those islands, collect needles for her. So we didn't know about this genetic structure differentiation of those populations at the small scale until about 5 or 6 weeks ago. So one of the first things that we're interested in thinking about is learning about the potential phenotypic differences among those, or at least trying to quantify it at some point in the future. But for now, we don't really know much.
You did say this was preliminary or recent results.
Yeah, well, you know, when we went out to collect the data, or when Lanie went out to plan the collecting of tissue for the sequencing, we were, you know, prioritizing in our minds being able, because these are, for these pines, at least Muricata and Radiata, we wanted to be able to sample every stand that exists, or at least some, you know, something close to that. So we were putting our energy into thinking about getting material for DNA from all possible populations, and less so on quantifying phenotypic variation. She has quantified a little bit of variation in SaVAtree up the coast in Muricata, and we still need to look at that a little more carefully. But we don't know anything about what's going on the island.
Okay, well, that'll be exciting to explore in the future. Okay, and then I'm gonna stop imposing on everyone after this final question, which is for Emily. And I was curious about this as well. So you've looked at genetic structure and the history of range shifts in your species, but how exactly does it relate to, well, here it says foundational knowledge, but I would say, you know, conservation and management, and especially with you know, connecting that back to the disease.
Yeah, absolutely. Because, you know, most butternut populations are affected by butternut canker disease. So there's not really a lot of remaining populations left. So it's kind of, there's like a few things we're thinking about. One of which is that the New Brunswick butternut populations are determined to be so distinct by our microsatellite markers and due to these examinations of past range shifts that they might have undergone being in certain conditions that are different for so long. We might need to protect them as a separate unit for conservation. So like in conservation groves, making sure that we're not planting them together to produce seeds because they might be maladapted to certain locations. In general, we also found a really big, you know, just specialization on mesic soils and very cold environments. And that habitat may be disappearing where they're currently planted. So when we're thinking about planting those individuals back out into the wild, because there's a lot of restoration efforts going on right now, we might need to start planting them in places that are not where they're currently found and what might be suitable for them in 50 to 100 years. And then sort of finally, we know genetic diversity is reduced in the southern and northern areas. So then we need to think about ways that we maybe should supplement those populations so they don't, you know, just go extinct after we plant, you know, individuals from those areas out. So there's a lot to keep in mind.
Yeah, for sure. Okay, well, I just want to thank all of the speakers for the fantastic presentations and Thomas for joining us during this live Q&A session.
Yeah, sorry I was a little late.
No, you were great. And also to all the attendees for the great questions. So I hope you guys enjoy the rest of the conference and thank you very much.
Thank you.
Thanks. Have a good day.
Thank you.